Before we start, are there any questions? Yeah.
Is it possible to post the lecture slides like right before class starts so we can pull them up while we take notes?
It is, of course, it's possible, but I don't want to do that. There's two reasons. I know. There's two reasons. One is I'm literally working on them right until I start walking here. And the second reason is it's better for you this way. Now, students don't like that answer. And when we get to the memory, I'll try and convince you of how wonderful this policy is, how much better it is for you to actually write things down yourself. But still, I know a lot of people want it anyway. Are there any other questions? Okay, today we're going to talk about sensation and perception together here. I'm not sure exactly how much we're going to cover today, but it's all kind of together, just broken up artificially across four slides. So today we're going to start by talking about, actually, one second, I got a lot more. Okay, so we're going to talk a little bit about sensation. But just before we start, I'm just going to introduce these terms. They're interrelated. So there's a lot, again, these kinds of terms, especially these high-level terms, are hard to define in a way that would satisfy every cognitive scientist, but let's just give it a shot here. So the idea of sensation is that you're really usually talking about the operation of your senses, the information processing that takes place in your senses, and the way in which that information is then transmitted from your senses to other areas in your brain. You can also include in sensation the early experiences of the sensory outputs. So even if you're looking at a picture of your mom, if she's wearing a red brooch, you can think about the redness separately from the fact that it's a brooch. In that, you can think about that object separately from the fact that it's your mom. So this is why these terms are a little bit hard to define, because the actual line between sensation and perception is pretty blurry. And in fact, the more research that's done, the more it looks like these things might be really sort of a back and forth combination of things that sort of happen in parallel. And where that line is a matter of current research. In our department, someone who's really doing an amazing job at the forefront of this kind of work is Jason Swaha. So maybe you can take one of his classes or check out the research he's doing. Anyway, traditionally the idea of sensation is the operation of your senses and the very early sort of experiences of that sensory information. And then perception would be somewhere downstream where you're talking more about combining those sensory experiences into some sort of understanding of what it is you're sensing. We used to, in fact, when I was in grad school not that long ago, we used to think of these things as quite separate, where we would call the sensation this bottom-up processing, meaning you would really go from the outside world, the information coming into your senses, and really just talk about the operation and the output of your senses. And then separately, you would think about this top-down processing, what your expectations were, what your knowledge base was, your experience was, and how that interacted to give you some sort of experience of what it is you were seeing or hearing or touching. And so again, it was seen as if the senses would feed information to the brain, and then the brain would then put that information together to perceive it. But now it seems as if the interaction between your sensory system and your perceptual system, your information gathering systems and your sort of understanding systems, they seem to be operating together in a much earlier way than we thought before. So anyway, what are your senses? Now you actually have a lot more senses than this. Some people don't even think we have this many. Everybody knows eyes, ears, nose, mouth, sight, audition, smell, taste. You also have a sense of where your body is in space. Yeah.
Sorry, do we do the roll call thing every time?
No. Just the first 2 days. That's just to see if there's anybody who enrolled who doesn't want to be here. Oh, by the way, if you were trying to add the class, this is unusual, but that process yielded no drops at all. So everyone either showed up or emailed me and said, please don't drop me from the course. So unlike previous quarters, that yielded no open spots for people that were on the wait list. So if you're on the wait list, you're still just waiting for someone to drop the course. All right, so census. So everybody knows about the standard 5, standard 4, depending on what you learned, but you actually have a lot more census. You have specific census that lets you know whether your stomach is full or not. There's all kinds of internal senses that we don't think about or not really conscious of in our everyday life. But your body has lots of different sensory systems. But anyway, the standard ones that we in psychology spend most of the time studying are your ability to see, hear, taste, smell, and your sense of touch. The vast majority of work in cognitive is on your visual stimuli. The vast majority. Maybe next is auditory stimuli, not maybe, definitely next is auditory stimuli. And after that, you go way down in percentage. You might get a few people doing touch, like your sense of touch. In fact, I haven't seen that much lately, but there's some. And then smell and taste are even more rare. You need more work in these other sensory systems. Right, the last bit here is just pointing out that your sense of touch is actually multiple. Where's my mouse? Your sense of touch is actually multiple separate capabilities. So you can obviously you can tell if your brother's punching you in the arm. I have 2 boys, so that's the first thing that pops up. But you can also sense warmth, cold. And you can also sense pain. These are separate systems. All right. So what do I want to say about your sensory system? Well, one of the things to say is you could spend your whole career studying one of these. And in fact, you could spend your whole career studying one tiny bit of one of these senses. These are really big topics each, and we have classes on some of these topics. For example, we have an entire class on, I think it's visual perception, but it includes sensation as well. We used to have a class on auditory, but I don't think we have that anymore. All right, so instead of going into detail about each of these systems, we're going to go into a little bit. Instead of just focusing on the systems themselves, let's talk a little bit about some things that are common across sensory systems. And one of those things is this principle of transduction. All of your senses have the ability to convert physical stimulus energy into electrical changes in neural receptors. So all the stuff that we interact with in the world is all some form of energy. So whether it's light, pressure, sound waves, you convert this energy in the external world into some pattern of neural firings, because it's got to ultimately end up with neurons firing in your brain, or else you won't be able to process it. So all your senses operate in the same way, in the sense that they take some kind of physical energy, and they convert that into a pattern of neuronal firing. Now we learned about the neuron before, and so we talked about how some neurons have receiving ends. There it goes. So some neurons have this receiving side, and they receive signals from other neurons, and those signals if you get enough of them, can eventually cause that neuron to send a signal to another neuron, and so on and so forth. Well, some neurons, some cells in your body are very much like neurons, except that one end of them is not evolved to interface with another neuron. It's actually evolved to interface in some way with the outside world. We call those receptors usually, but they're basically just a fancy neuron where one side is sensitive to some kind of energy, whether that's pressure, or something like light. So we'll talk about that in a sec. But let's first talk about their sense of smell, not their sense of vision. So one interesting thing about your sense of smell is that the specialized receptors, the specialized neurons that have one end sensitive to some sort of energy, is they actually stick right down outside of the top of your nose. Of course, you have no personal awareness of this because it's really in there. And so at the top of your nasal cavity, poking right through the bone, are these little tiny, these photoreceptors, and in particular, these little tiny hairs at the end of those these olfactory receptors, so for smell. And they have these little tiny hairs that poke out of the ends of them. And those are the ones that actually poke through your nose and into your nasal cavity. They're called cilia. They're little tiny hairs. And those cilia are sensitive to various different kinds of chemical ions. Some of them are actually connect. Well, let's not get into the chemistry of it. But so some ions, whether certain ions cause those receptors to fire. Some of those receptors actually only fire for various kinds of combinations. So various chemical compounds. But they're sticking right through the top. Yeah. And underneath those is a mucous membrane. Yeah. There's a mucous lining that is right under them. And so what happens is when you smell something, particles of that thing that you're smelling walk through the air into your nasal cavity, and if you sniff hard enough, the arc of those things will go towards the top of your nasal cavity and have a chance for particles of that substance to lodge themselves into the mucous lining, and then sort of float their way up to where the little hairs are. And if those particles go through the mucous lining up to where the little cilia are, if they're the right kinds, they can then cause electrical firings in those receptors. And as you can see, the receptors look just like neurons. Yeah, so these look like neurons, and essentially they are. They have a different name because what would be their dendritic end is really something that's evolved specifically to interact with the outside world. In this case, these neural receptors only fire when in the presence of some kind of a chemical compound or some kind of chemical compound. Let's say. We can't smell everything. There's plenty of things that to us have no smell, so we can only smell certain things. And if they happen to make it into your nose and up to the top, and they make it through the mucous lining, then there's a chance that you might smell them. And what does that smell mean? Well, it means that signals have to be sent through this olfactory bulb is just this really thick sort of cable of these smell receptors that then go to your brain. And so in order for you to smell something, this process has to happen. When I was in Psych, when I was taking intro Psych, I remember being quite grossed out by this. Because I grew up near farms and there's lots of horses, and all of a sudden I realized that the only reason I was smelling horse shit was because it was literally going into my nose and sticking in the mucous lining and then interfering with I don't know, that was weird to me. In fact, I've looked at the world very differently since then. It's like, I thought it was just some kind of a at a distance signal that I was interpreting. I didn't realize it was actually going into my body. I thought that was gross. But apparently that's how it works. So those receptors take that information, and then on their terminal buttons, they're connecting to other normal what we call interneurons. The normal neurons we talked about last time, the ones that mostly talk to other neurons. Well, at some point you have to get the signal to your brain, and so the specialized smell receptors, when they fire, they cause normal sort of interneurons to fire, and so on, all the way to your brain. Where do they go into your brain? Well, there are various projections from the olfactory bulb. And so that information goes to lots of places. One is your hippocampus. So there's a pretty direct connection between your sense of smell and your memory, or at least a part of your brain that creates new memories. Some projections go to the thalamus, which does some early processing. The same is going to be true when we look at vision, and then sends signals on to different parts of your brain. One area is your hypothalamus, which regulates emotions, drives, and pleasures, and another is your amygdala, which, as this here, includes things like hunger, sexuality, fear, and panic. The amygdala, most people know the amygdala for its influence on the processing of fear and panic, but it also has these other features as well. Okay, so that's your sense of smell. That's just a tiny, tiny first broad strokes of your sense of smell. And what I'm focusing on here is not everything there is to know about your sense of smell, but just trying to have you see the way in which it translates something physical in the world, in this case, movement of chemical ions, into neural patterns of neural firing in your brain. All right, so on this same idea of transduction, let's look at how the tongue does it, okay? Surprisingly, it's very, very similar. So one thing to point out is that historically, not that long ago, I mean, I've definitely taught with textbooks that had pictures of the tongue like this, and they would say, if something gets on this area of the tongue, it will be perceived as sour. If it gets on this area, it's sweet, and so on. That turns out to be wrong. That research could not be sufficiently replicated, and it turns out that you do have different receptors that specialize for things like sweet, sour, salty, bitter, but they are distributed all over the tongue. This sort of this sort of specific mapping like this, it just turns out isn't true. So your tongue is just this field. One second, let me see. I thought I had a picture of this. I'll come back if I don't. I don't. Actually, I don't believe it. One second. It must be nice. Fine, but I will describe it to you with words. Okay, so your tongue is this field of little bundles, which are essentially a specialized kind of receptor. It's very similar to the ones in your nose, in the sense that the part that sticks out into your tongue is actually these little tiny cilia. The difference is there's also, in addition to the cilia, there are also little grooves that chemical ions can fall into. But let's just, at this point, just say your tongue is covered in a field of specialized receptors. They look different, they have different morphology than the ones in your nose, but essentially, the business end are these little cilia that stick through. And again, I'm oversimplifying the structure, but let's just go with it. So these little cilia stick through, and just like and it's also covered in saliva, in a layer that's fortunately sticky. And food goes into your mouth. Food is made up of different chemical compounds. Some of those compounds, when they interact with the receptors in your tongue, they cause them to begin firing above baseline. And again, that's how it converts this physical stuff into patterns of neural firing. You have taste receptors, like it says here, for sweet, sour, salty, bitter, but also for hot and cold, okay? We're going to talk a little bit more about those different qualities, but for a moment I just wanted to point out that the signals coming from your tongue take a different route to your brain. They actually go to your brain stem. Instead of going from your nose basically this way into your midbrain, they actually go to top of your spinal column here. And there's a little, and so anyway, there's one group has one sort of main connection, and it's the back of your tongue actually has its own separate connection. And then this is actually showing something that's not related to taste, but this is the, I think it's called the epiglottis, yeah. And that's actually has to do with you not drowning yourself in your Diet Coke. So your ability to know whether something's about to go down your windpipe. So anyway, these go to your spinal column, and then they are then projected through your thalamus, your hypothalamus, projections to your amygdala, again, fear, and sex drive, taste, things like that. It goes to your hypothalamus, very similar areas that your sense of smell goes to. And in fact, in terms of the relationship between your sense of smell and your sense of taste, we are not always aware of the relationship between them when we're smelling things and eating things. And in fact, there are some foods that actually have no comparable receptors on your tongue, or at least the parts that we think of as unique about those foods. So for example, something like chocolate, even though you can of course taste the sweetness of chocolate on your tongue, but the chocolatey part that we enjoy and love about chocolate, that's mostly your sense of smell. And so there's lots of foods like that where it's either totally your sense of smell or not your sense of smell at all, only with your tongue, some combination of the both. We are really not aware of which one is which unless you're trying to eat something that you really like when you have a cold, and sometimes you will notice that some things taste relatively the same as before, but some things you can't taste at all. Yeah?
If you're smelling something that you're eating, is it going through your nostrils or is it going through like the back of your mouth into your nasal?
It's probably both.
Okay.
I mean, yeah, in fact, you could actually experiment on that yourself. But yeah, it's usually some combination. It depends on what part of the process you're on. Sometimes if you're chewing not the process, but if you're chewing something that involves your mouth opening,
Yeah.
but some things you might, if you have like a cough drop or something in your mouth that's closed. So yeah, there's definitely an opening back there, just as there's one in the let's see, what's the point here? Oh, well, I guess the idea of this slide is to just point out that there are some, evolutionary psychologists have some ideas about why we might, have evolved certain kinds of taste receptors. And so there's no evolutionary story here for salty, just pointing out that we have lots of systems in our body that are sensitive to the presence of sodium ions. But with sour and bitter, in nature, oftentimes things that are sour are things that you are decaying and that you don't want to eat, lest you get sick. And so the idea is that maybe the sour receptors evolved to help us avoid eating things that are decaying. Obviously, you add some sugar and some citrus flavor, all of a sudden you've got some candies that we know and love. But just sort of this non-sweet kind of sour, just in nature is often is a sign that something is decaying. And the same thing with bitter. Obviously people drink coffee and consume other things that are bitter. But again, in nature, oftentimes if something's bitter, that signals the presence of alkaloids, which are often associated with things that, again, you don't want to eat. So whether you can take these to the bank or not, I can't really say, but there are some ideas about why we can sense certain kinds of chemical compounds and not others. There are similar stories for our sense of smell. There's similar explanations for why we can smell certain things and not others. The other thing to point out is that those receptors are relatively dumb in the sense that they're just looking for certain kinds of chemical compounds, and in the presence of those compounds, they will fire a baseline. We constantly fool ourselves by consuming things with certain kinds of compounds that actually trick our taste buds. So for example, if you put menthol, which is a common thing in something like these Listerine strips, but also things like gum and cough drops or whatever. If you put menthol on your tongue, it will activate receptors that are usually activated for things that are cold. So people actually feel like their mouth is cooling off when you have menthol, although no temperature change whatsoever is occurring. And again, if you've got a kitchen thermometer or something, especially one of those fast thermometers, you can test this out yourself. There's no temperature change whatsoever. And the same thing is true for the chemical compound called capsaicin. If you put that on your tongue, it will cause an increased activation in the receptors that usually respond to changes in actual things that are actually hot. And it will make your mouth feel hot. And this one is pretty compelling, as you know. If you eat enough ghost pepper potato chips from Trader Joe's, like I do. Anyway, if you eat enough hot sauce or something, you will literally feel like your mouth is on fire, and it not just feels like it's hot, but you will enter a crisis mode where you really feel like you have to put this thing out. You have to do something to change it, even though it's not actually hurting your tongue. Nothing's burning. Your brain doesn't know any difference. The receptors in your tongue have sent signals to your brain saying it's hot, and as far as you know, there's a crisis now. And so there's no temperature change at all occurring in either case. There's a hand somewhere. Does someone have a question?
You answered my question.
Okay. Okay, so again, the point here is we're thinking about the common ways in which they turn physical things into patterns of neural firing. And your tongue and your nose, they basically do that in nearly the same way. That is, you have these cilia that stick out, and they actually, when they're in the presence of various chemical compounds, they change their firing rate. They change, and those receptors then link to other interneurons, which then make their way through the brain, and eventually to areas of your brain that allow you to process things like, what is this taste, or is this something I want to keep eating? Well, the same thing happens in your ear. The same general principle occurs in your ear. Our sound is just compression and decompression of air, or some other substance, but typically air. Well, we've evolved this system in our ear to convert changes in sound waves, not changes in sound waves, which again is compression, converting sound waves into neural firing. So this physical thing in the world, we have a sense that convert that physicality into patterns of neural firing. And so in your ear, there's this canal, and it leads to these little bones. There's 3 tiny little bones. The hammer, the anvil, and the stirrup. I usually have to have this little mnemonic to help me. Anyway, this hammer, anvil, and stirrup, and basically they just vibrate. The first one vibrates. This one is actually exposed to the outside world. It vibrates as the sound waves come in. It causes the anvil to vibrate, and that causes the stirrup to vibrate. And ultimately, there's this little purple arc here. There's this tiny little membrane there, almost like the membrane on a speaker, that vibrates as the sound vibrates. Now, why we didn't evolve a system where this structure is right there? Why do we have to have the bones? I don't actually know. I don't have an evolutionary story for that, or even a mechanical story as to why that's better. So I don't actually know. But ultimately, the sound waves are transferred to this little membrane called the tympanic. What is the point of all that? Well, that membrane is connected to this structure here called the cochlea, which is basically just a tube. It's all wound up, but you can think of it just as a tube. And inside that tube is liquid. And so if you does anybody have or grew up with a fish tank in their house? What happens if you start banging on one end of it, just in a rhythmic pattern? What will happen inside? What was it?
Water moves.
Right, the water will start moving. And in fact, if you've done it long enough, especially if you keep a regular pattern, the water will start to move in a regular way. That pattern, that phasing will be transferred into the liquid in the water. What happens if you have grass at the bottom of the fish tank? What does it do? Yeah, there you go. So some people are showing me with their hands. Right. That's exactly right. Correct answer. Well, interestingly, that's how your inner ear works. There's this tube, and again, it's rolled up. Let's just think of it as a long tube, and right in the middle, there's a layer that has a bunch of little cilia on it. Again, a bunch of little hairs. But these hairs don't increase their activation when in the presence of chemical compounds. They increase their activation as a function of how much they are deflected. So just like grass at the bottom of the ocean or in your fish tank, as sound waves start hitting the tympanic membrane, so basically hitting the end of it, the cilia inside start to move around in phase with the signal that's coming in. So this movement of air is translated into deflection of these cilia, which themselves, each one of them is associated with a receptor, which changes its firing rate depending on the way in which those hairs are moving around. So again, all these senses, your sense of smell, your sense of taste, and your sense of hearing, surprisingly, all work in basically the same way. They have different contexts. One's on your tongue, one's in your nose, and then one's inside the cochlea here. But the way they operate is roughly the same. Sort of the technology, if you will, the structure of them is really similar. We'll see something different when we look at how your eyes work, but these senses are all very, very similar. Oh, wait, I have another picture for you here. Yeah. The signals that come from here, this is just showing that it goes through your temporal lobe. So your primary auditory cortex is your temporal lobe, but your auditory nerve goes to your thalamus and other areas as well. But yeah, the primary auditory cortex is in your temporal lobe, in particular your left one, but actually both. Okay. I want to talk about another principle, which is neural coding, okay? So you can't, it's not like we have one receptor for loud sounds and another cilia for soft sounds and another cilia for trumpets. Some way we have to, your body has to convert these patterns of electrical, neuronal firing into the qualitative qualitative experience that we have of things in the world. So somehow the stimuli that we engage with have to not just cause our receptors to fire up a baseline, but somehow our brain has to use that information to understand what it is we are seeing, hearing, and tasting. So, one thing to remember is that, again, we've talked about this before, but just in terms of neurons are firing at some baseline level. And they can be, if they get lots of signals from other places, either other interneurons or from sensory receptors, they can increase their firing rate above baseline. And by firing rate, we're just talking about how many action potentials that neuron generates per second. Well, with something like sound, then you can't just say, well, the cilia will move around in the presence of sound, and they won't move around where there is no sound. That's not enough information, right? So we need to be able to understand other things about the sound that's coming in. So for example, even just to describe a simple tone, or just one key on a piano, which is actually, if you know anything about music, I've just made a blunder. So one key on the piano is not a simple tone, but anyway, fine, let's just say it is. So just in terms of one note on an instrument, these are not simple, they're actually layers of different simple tones. But in order to represent one simple tone, you need to be able to represent not only the frequency of the sound wave, so a high-pitched tone will have a faster frequency. A low-pitched tone will have a frequency, a peak less often. But you also have to represent loudness. How loud is the tone? So the amplitude, how large the deflections are in the wave, will indicate, is a measure of how loud the sound is. So big deflections, loud sound. Little deflections, low sound. So again, this is simplistic because there's all kinds of other things we have to code for. Like I said, you have to know whether it's a trumpet or a piccolo. But even in terms of simple sound, we need somehow deflection of cilia to lead to our understanding that this is a high tone versus a low tone, or which tone it is, as well as how loud the tone is. Somehow that information has to be coded in your brain when the only input is deflection of cilia. In your ear, one of the ways this happens is not so much, well, let me say, so in terms of loudness, how loud something is, that's again, this is all simplified. That's more or less a function of just how much deflected the cilia are, okay? So if you were to just tap on the side of a fish tank, well, there's some threshold, you have to figure out what the lowest threshold is, but at some point, you might cause the grass to move a little bit. But if you were to slam on the side of it, and your mom didn't tell you to cut it out, you would notice that there are much bigger deflections. As the water moved more around, the deflections would get bigger. Well, in your ear, that's more or less how amplitude is coded. So bigger movements are perceived or experienced as louder sounds, and smaller movements are experienced as lower sounds. So that one's easy. But what about pitch? Well, things like pitch what note it is, are I should just say, let me just say this. We really need more people to be interested in the cognitive psychology of hearing. There are actually several theories for how we do something like distinguish a trumpet. There's even multiple, distinguish a trumpet from a flugelhorn. So not even trumpet versus a flute or a guitar. There are theories for how we do it, but this is one area where there are multiple good theories, and people have not yet figured out which one is actually correct. So it's one of those areas in cognitive psychology that seems like it's asking a question that a lot of people will want to know the answer to, but for science, it's really not figured out the correct answer. Yeah.
Why is there not so much research in like hearing and some of these other? Is it just like not enough money, or?
I don't think it was a funding issue. I don't think it would take. It's not like it would take lots of expensive equipment or anything. I just think historically it was just easier to show people visual stimuli and ask them, was it this stimuli or that, this stimulus or that stimulus? Be able to present those stimuli, control them. Historically, the equipment was just easier to come by. Now we have pretty good equipment for presenting an auditory stimuli, but even today, even in 2023, the vast majority of cognitive studies are done with visual stimuli. I just think it's easier, people just find it easier to reason about. Here's another important difference though, is it's not just lack of respect for auditory. With visual, your ability to detect a visual stimulus is more or less a discrete judgment. So the way the cells in the back of your eye work, you can tell without any other reference at all whether there's a light on or not, or whether a stimulus is here or there, or whether a stimulus is a G or a T. So you can do that by only consulting that light energy itself, or that position on the screen. You can do it fast, and you can do it very precisely. And again, with only that information, so that leads to it being really fast. With auditory, unless you have perfect pitch, and probably not even then, most people when they make auditory judgments, like press this button if it's a high tone, press this button if it's a low tone, those are relative judgments. You have to make that judgment with regards to some standard. So the processing that's required is slower, and it involves multiple regions of your brain. It's really that slowness that left it as a bad candidate for lots of kinds of research. So part of the preference for visual stimuli is just how easily we can distinguish one stimulus from another, even when they're very close together. So you take two visual stimuli, it can be quite different stimuli, or even quite similar, and you can put them right next to each other. And people have no problem telling you what those two stimuli are, or only responding to the second one. But if you have two tones that are even relatively far apart, people have to do a much more complicated judgment. So that's in the psychology side. There are people that just, from the physical, doctors and people in the physical medical kinds of sciences who study how the ear works. We understand the physical aspects of it. But in terms of answering these questions specifically, how do we distinguish one instrument from another, or even one quality of an instrument? Instruments can make all sorts of sounds. We can perceive them quite easily. How do we do that? It's not that there's no work on it. But there are multiple theories and just not enough work to have sort of grind through them and figure out which one is correct. There are multiple theories for how we actually achieve that. That actually fit the data more or less. But which one is right, we don't know. And they're quite different. Okay. Yes, sorry. Go for it.
Okay. That's kind of interesting because I'm a musician and I sing and I play through instruments and I am able to tell a pitch and tell what note is a middle C or a low C or a high C. And there's even moments where I'll hear a song and I'll remember it, it'll be in the back of my head and I'm able to play it without even looking up if those are the right chords or not. So it's kind of interesting.
Well, do you have perfect pitch?
I do.
Yeah, so you have a superpower. Most people can't do what you can do. So most musicians, I'm also a musician, and I'm sure there's tons in the class. Most musicians, if you play them a tone, they may be able to tell you what it is if they're have enough experience. But they do it by doing sort of a relative judgment. It's called relative pitch. So most musicians most people on the planet are like that. They're in that boat. There are people that have perfect pitch. There's some debate as to whether you can teach it. It seems like maybe you can teach it, but a lot of people are just born with it. And whatever's going on, it works differently. Anybody? Oh, you had a question.
Yeah. This is a random question. So sometimes when I'm told something, I'll say huh, and then understand half a second later. Is that just the signal going straight?
Let me think for a second. Did we cover it? I get a lot of interference with this class and the other class I teach. I'm trying to, do we cover that? This class or not?
Because I only have this to me a lot.
Okay, I don't think we covered. So there's some research on this. There's actually multiple reasons why that can occur. Some of it can be attentional, but some of it can actually be auditory. The interesting thing about your sense of hearing, you do cover that. I'm already doing it. So the other thing about your sense of hearing is that, it plays out over time. Again, these things literally have to move around. They actually have to move around before you can tell what's going on. So it takes more time. Sometimes people ask for a reset, you say, whatever it is. Even though a moment later, have you ever had that experience where a moment later you know what it is? Let's say you say what'd you say, and they don't answer. Sometimes you go, oh yeah, I know what it is. People have that experience. So that's why there's multiple reasons. Some of it's impatience, and you would have gotten it before. Some of it's an attention where you didn't get it because you were looking somewhere else or in your head thinking of something else. So there's multiple reasons why it happens. I talk actually about this a little bit I teach a class on lying and deception. So we actually go into some detail on it there in ways, because it can be used in some ways as a stalling tactic. Go ahead.
For something like the Doppler effect, where you're listening and
from like the like pitch or whatever,
Mhm.
you're able to like tell like distance and like
Mhm.
depth perception. How does that like does that because you can also do that like with your eyes and stuff like that, like with depth, like distance and stuff. Does
Well,
that, like, draw
those are very different. Those operate in very different ways. With your ears, because you have two of them, a sound has a particular location. And so because we have two ears, they come into your ears at slightly different positions, slightly different times, I'm sorry. And so we use the relative time that they hit our two ears as a judgment of where they are. And it's just simple geometry.
Oh, so it's not like going to a different like receptor for like depth perception or something, right?
No.
It's just all like within the audio.
Yeah. No, the way you're working your vision is totally different. All right, so another principle is that your sensory systems interact, you can even say operate, across space and time. That sounds like science fiction or something. That sounds right. All right. Oh, here we go. I don't know why that did that. So yeah, what you sense now in any given place depends on what you sensed a minute ago and what is happening around you. All right, so there's an eye. There's lots of structures here, but the most obvious ones is your pupil. The white part is the sclera, and the colored part is the iris. One thing to know about your sense of vision is that light comes into your eye and is then focused by your iris and your lens. That's good. Keeps our vision from being too blurry. But what's interesting, one interesting thing is that when the light comes into your eye, it's actually flipped upside down. So the light comes in, and it then ultimately gets projected sort of on the back of your eye where your photoreceptors are. But the signal that gets projected on the back of your eye is upside down. It's flipped upside down. We don't know, of course we don't experience the world as upside down, but that's just because we're used to it. Your brain is just ignoring the fact that it's upside down and is just literally, essentially writing it in how you perceive it. You might be skeptical of that, but it's actually pretty simple to go on Amazon. You can buy prism glasses. These glasses that essentially have a prism on them. If you put prism glasses on, it will flip the world upside down, okay? And it will be very difficult for you to live your life. But if you try it, if you keep them on for, I don't know exactly how long it takes, 5 days, 4 days, or maybe a week. If you actually just fumble through the world, as you will if everything's upside down, in about a week's time, your brain will just flip it again. It'll just go, okay, so this is the new up, fine. It'll flip it. All of a sudden, boom, everything looks normal. Then you're gonna have another tough week when you take them off, and it'll flip it again. So your brain is just doing this, it's just flipping it, yeah.
Would that impact would that give you a lot of vertigo and just like a lot of...
Oh, it'd be terrible. Yeah, it'll give, yeah. I mean, actually, vertigo is a specific thing that's happening. And so, yeah, if you have any sort of tendency to vertigo, it will definitely exacerbate it. But it won't give you vertigo. Vertigo has to do with crystals and your inner ear. It's a very weird thing. If you're interested in vertigo, read about it. And just be prepared for the word crystals to be a serious subject about it. Yeah.
So like, is the lens like it appears up there like an ovular shape, or does it have like a convexity to it? It's like, you know when you look at a spoon, if you look at the inside of the spoon,
Yeah.
you're upside down. If you look at the back, you're right side up.
It's like it has that shape.
Okay.
But there are also muscles that can move the shape around. So I'm definitely doing broad strokes on how the eye works, but there's a lot of detail there, and yeah, it's like two spoons together, but they're transparent. That's part of the reason why it gets flipped. All right, so what's the point? Well, light comes into your eye, and there are specialized photoreceptors in your eye, which is how we convert light energy into neural impulses. But they are at a specific location, at least the best ones, the most precise ones, are in a specific location. And it's called your fovea, and it's that little teeny tiny area that red line is pointing to on the right side. Here's a picture. Some of you may have taken a picture like this here if you've gone to an ophthalmologist. older people like me have to do it to see if we have certain kinds of eye diseases. But anyway, you can take a picture of the back of your eye. And if you do, the fovea is a really tiny area. And when you're looking around the world or trying to read or trying to really understand something visually, what you're doing is trying to get your eyes lined up so that the light that's coming in of the thing that you care about is right on your fovea. But it's tiny. And so it's, you have to actually move your eyes around to do this. Well, interestingly, so there's a field of these photoreceptors, and they're all sitting there and they're all interacting with the light, but eventually they have to send their signals to your brain, right? Well, what happens is all the terminal ends of these photoreceptors, the terminal button side of it, they all sort of go to a bundle of fibers. It's kind of like you've got a big giant cable, and they all sort of converge at that cable, and then that cable goes out of your eye into your brain. And because all these photoreceptors have their axons going through this one part of the back of your eye, there aren't any photoreceptors doing any work at that part of your eye. And so we call it a blind spot. And you can find it's another thing that your brain handily does for you. There's a hole in your vision right now. Like literally there's a hole, because there are no receptors there. But you don't experience it, because your brain is literally taking all the information around that area and just kind of stitching it together. Your brain does lots of cool stuff like that. Well, anyway, you might not believe me, so you can find your blind spot pretty easily. Just hold your thumbnail out in front of you, right in the center. It's going to be hard because you're going to move your eyes, but keep your eyes where your thumb is. Close one eye, and then actually close your right eye. Keep your thumb in the middle, and then keep your eyes where your thumb is, and move your thumb to the left. It may take you a minute if you have a tendency to look. Eventually, you'll find a spot where you can't see your thumbnail. It's right there. It'll just literally disappear. If you can't see, you got to cover one eye. You have to cover one eye. And you got to do it slowly. You got to keep looking in the center while you move your thumb over. But you do have to cover one eye. If you're holding your right eye, you move to the left.
That's crazy.
You think I'm not able to find it? It's there. All right. So let's talk about these receptors.
The new vision ratings of 25.
So your brain, you have two different kinds of photoreceptors. And the photoreceptor is, again, a specialized neuron where what would be the dendritic end, instead of being dendrites, is a specialized receptor that responds to a certain kind of energy. Instead of a certain kind of chemical compound, these receptors respond to certain kinds of light. We have two kinds, the rods and the cones. The cones are the ones that are, some books say that the cones are only in the fovea and the rods are in the periphery. That is not the case. That's been not the case for a while, although I've seen it in modern textbooks. But the highest concentration of cones are in the fovea, that little teeny tiny area. Those are the receptors that take the most light to do their job. They're highly sensitive to color. They're also more precise. But that precision is not necessarily because of the individual receptor. But anyway, the highest acuity, the highest precision, the best color vision is in the tiniest area here at the back of your eye, the fovea. Rods, on the other hand, they're distributed all over. They're just less dense in the fovea. They can operate under low light, but they're not so sensitive to color. Yeah.
How people say eagles have the best eyesight and can see a few miles. Does that mean they have smaller, more concentrated sight cells? Or
I understand the logic there is sort of about yeah, more, it's almost like a camera having a bigger sensor or something, like more pixels. What's remarkable about an eagle's vision is not necessarily how their eyes work, it's how much of their brain is devoted to vision. It's like a third. Or maybe even more, depending on some birds actually have better eyesight. So yeah, a lot of animals like that, even animals that have a better sense of smell or whatever. Like your dog's sense of smell is like 40,000 times, 60,000 times better than yours. Anyway, sometimes it's about better sensory systems, but the biggest, I think, the most variance is taken up by the fact that they have a much bigger part of their brain devoted to that thing. Yeah.
Aren't there animals that have like different kinds of cones as well? So like the
Yes.
mantis shrimp.
So exactly. So you can have, some animals have multiple different kinds, more than 2 kinds of receptors. Some animals, we are sensitive to red, green, and blue. We have receptors that, some animals have receptors that are sensitive to other things. Some animals have eyes that are broken up in a way that causes a lot of redundancy. There's all kinds of technical differences in the way that the eyes work that lead to those better senses. And in fact, some animals, the only reason they have a better whatever is because of differences in the sensory system. But I was just saying that particular example, and a lot of examples like that, like why some animals have a better sense of smell or better sense of vision, those really have to do with how much processing power they've evolved for that sense. But you're right, there are also different creatures that have different types of eyes, and sometimes they're better than ours from one perspective.
Is there a reason why those percentages don't equal 100? Is there like 2% that precedes other colors?
That's a good question. I don't know. Maybe I lopped off some fractional parts? I don't know. No one's ever noticed that before. I just assumed they did. Okay, I'll look into that and fix it if necessary. Yeah. Yeah.
When it comes to color blindness, does that have to do with like the tones, or does that have to do with connecting to the neuron and registering the color, or does it have like what happens when you're colorblind?
What's the answer to that? Isn't it more of a brain thing, or is it an eye? With color blindness, isn't the issue in your brain, or is it in your eyes?
It's in your eyes. It's in the eyes.
It's only in the eyes?
Well, there's different types of color.
Right.
It's from Nick's previous class. I took his perception class. It's more so that you only have 2 of the 3 cones, and it depends like which cone you're missing, and it is based on like the eye of it.
Okay.
You're just born without a specific cone, which then correlates to what color blindness you would then experience.
Okay, I did not know that. I know you're surprised. You thought I knew everything. Okay. Oh my God, I gotta keep doing this. It'd be okay if I did my password was only 40 characters long. Okay. So we've got these rods and you've got these cones. One thing to keep in mind, especially if you're familiar with your modern phones and cameras, is that you have a total of like 127 million dots, or points of, I don't know. Your camera and phone, they use a field of essentially receptors. And those numbers get much, much bigger than what's available in your eye. So 127 million sounds like a lot, but it's not. The phone in your pocket right now, or the camera that's in your bag, has way more receptors, way more sort of precision than the human eye. And this is only one way in which the current camera that you have is better than your eye. We'll talk about that when we get there, yeah.
What part of your eye is affected when like you don't have perfect vision?
When you don't have perfect vision, it's really about the lens and where it's actually focusing the light. So the light can be focused essentially behind where it needs to. It needs to be focused perfectly right on the receptors. And so it can be before or behind. And so you'll have trouble seeing without, and so your glasses are trying to correct that process. Make things line up right where they need to. Okay, so rods and cones. Why are they called that? It has to do with their shape.
I have one more question.
No problem, no
If
problem.
there's more red cones than green or blue, does that mean that we can see more shades of red, or is it?
It doesn't quite work that way. There's a lot of detail that I have to skip over here, folks. So and I'm not saying you shouldn't ask a question, I'm just explaining that. Some, like red and I believe green, those receptors are actually right in the center of your fovea, but your blue tends to actually be a ring around your fovea. There's all kinds of weirdness of how these things are arranged that influences the kind of colors you see. And later on we're going to talk a little bit about component processes, and those actually have an impact as well. So the number of reds doesn't translate directly into shades, partially because in order to get the shades, you actually have to mix in other colors. So it's a little bit more complicated than that. The other thing is that some, I forget the percentage, it's a fairly low percentage, single-digit percentage of women actually have an additional color an additional type of cone. And I can't remember what color it specializes in, but it is true that some women actually have better color perception than other women and men. And I mention that because, of course, there's the stereotype that's based on socialization. Some women are better at color perception just because of how the differentiating ways in which we raise boys and girls. We treat them differently, they do different activities. So if you remove those socialization differences, most cognitive differences disappear between men and women. However, some women actually do have additional cones that lead to much better color differentiation.
Do you think that has something to do with like why males have a higher chance of being colorblind?
I don't know the answer to this, so I just don't know anything about this. You could be 100% right. I just don't know. Yeah.
So I heard of this like theory that like basically every person could see the world like completely different in terms of color. Like but we would never know because we label them the same. Like when we're young, we're taught that this is red and this is blue.
Right.
But this is like very specific of certain colors that. So
I know it's a fun philosophical sort of idea, and I've definitely thought about this too. But we don't. I mean, yeah, we don't. We all see red the same way, and it's not. If it were something where, well, because of the way your eyes work, the same receptors are firing in the same ways when you see red. It's not completely an interpretive kind of thing. So you're not. I mean, this is a common thing that people think about, but that can actually fairly easily be tested. We can ask people lots and lots of questions about what they're experiencing, and you can really hone in on the different colors. And so maybe one person might, just because of the way their eyes work, maybe see things a little bit brighter. There can be small shifts in hue, but no one is looking at red and experiencing green. And again, even before science, you can just look at literature, poetry, art. You can really, it's fairly straightforward that we see the same. We have the same experiences when we see these different colors. But you can also test it scientifically. You can really answer the question.
This kind of reminds me of like the whole dress trend, where it's like the blue and black dress, and whether or not you see blue or black, or you see gold. What was it, gold and white?
They talk about that in your book, so I'm gonna skip that topic, but that's actually covered in your book. It is interesting. Yeah.
Just color blindness. The trait for, I think, red-green colorblindness is on the X chromosome, so it's genetic, which is why it's more prevalent.
That's interesting. So who asked that question about why? Yeah. So it seems like it's maybe genetic. That's Wow. Okay, let's move on. This is fun. Let's see, rods, cones. Right, so you might say, well, yeah, so when I first talk about rods, saying they only operate they operate in low light, they can't operate in low light, they're not sensitive to color, you may think those are not so good. But rods are awesome, even though they are not very sensitive to color, they are very sensitive to, they can operate in lower light, which is already useful. But they're really good at distinguishing patterns of dark and light. They're really good at processing motion. And so they are very useful. In fact, if you find you know how when you're in the dark, like if I go into my laundry room, which has a ridiculously bright light at night, and then I come out and I turn the light off, and now I'm in the dark, I can't see anything. You probably have situations like this as well. One thing you can do is move your eyes around and try and see things in your periphery, like really focus on looking at your periphery. And sometimes you can see more than you could if you're just trying to focus on full field vision. So we have these rods and cones. Actually, here's a little picture. This is an electron microscope. They literally are these shapes. So on the right here is an actual picture of rods and cones. Now, the strangest thing about the way these work in the brain is that they are sort of put together in the opposite way that you might expect. So your rods and your cones, whose ends, by the way, are light sensitive. These are literally the photoreceptive parts. Those are actually pointed to the back of your eye. So the light comes in from your lens and goes through these ganglion cells, it goes through these omicron cells, through these bipolar cells, it goes through the body of these, I missed the horizontal cells, through the bodies of the receptors, and then it gets to these light-sensitive parts. There are some ideas, I haven't seen anything that's obvious to me, but there are definitely some ideas about what would happen if you had it the other way. It has to do with light scattering. The way this works here is that the light comes through, and some of the light hits these receptors and gets processed by them, and then there's a layer at the very back of the eye that is very, very black. It's very dark. In terms of pigments, it's very, very dark, and it absorbs light really, really well. And that also is all about reducing the degree to which light bounces around. If light were allowed to bounce, if this have evolved in the way that light could bounce around in your eye, receptors would get activated even though that part of your eye, there's no light actually pointed at that part of your eye. So it's weird, but there's actually a good reason to have it this way. So the receptors, the light comes through all this muck, it hits these receptors, and then the signal goes back this way. And eventually, again, there's this sort of collection of fibers that all sort of go out of the eye into the brain. Let's see, where are we in time? It's okay. Okay. I want to talk to you about another, this is still using your eye, we're going to talk about another principle, and that is this idea of interaction in time. And it's just a general idea that we read before, which is that what you perceive now, right, at time, at one particular point in time, is influenced by what you perceived at a previous point in time. Your body might be your senses and your body and your brain might be in a different state than it was a millisecond ago, and that can influence how you perceive the same stimulus. So here's a little color after image experiment here. So I'm going to put this on the screen, and so what you should do, let me tell you what's going to happen first. So I'm going to have you stare at the center of one of these crosses, okay? You don't have to do it yet. And because you're staring at the center, it means that the upper right part of the back of your eye will only be getting red. And the bottom right will only be getting blue, etc. Okay? So what we're doing is we're concentrating a particular color of light on particular parts of the back of your eye. Why are we doing that? We're doing that because just like any cell, your neurons are just, and receptors are just like any other cell in your body. They're living things. They eat, they convert food to energy, they produce waste, they have, they're living things. And when they are processing, when they're firing above baseline to the color red, they get tired. Just like any other thing doing work. Any other living thing doing work. So by having you stare at the center and having red in only one part of your eye, the receptors in that part of your eye that fire for red, the cones that are specialized to fire for red, will get tired. They'll get more tired in that part of your eye than cones that are processing other colors. And there's going to be an effect of that. I mean, then have you look at something, and it's going to look very different than before I made some of your cones tired. So that's what we're going to be doing. So let's do it. So I want you to just pick one side, stare at the cross, and just keep your eyes there until I tell you. You can blink, but just always keep your eyes at the black cross, okay? Let's do that for a little while. Keep staring. You can blink, just keep, every time, just go back to the cross. Don't look around, straight at the cross. Now, while you're staring at the cross, please don't stop staring at the cross. I'm going to tell you what's going to happen next. So, I'm going to show you another grid, and that grid has no color on it. It's just, well, it does. It's just all gray, okay? It's just going to be 100% gray on the whole thing, okay? Just keep staring. Okay, I'm going to change it, and I'm going to show you gray, and you're going to see something other than what's on the screen. Okay, I'm going to do it now.
What?
So,
Yeah, it's gone.
okay, so here's what happened. It's sort of what I said before. But the new thing to notice is that this gray has middling and equal amounts of red, green, and blue. Okay, that's how you make that gray. And I've included, if you know anything about RGB colors, RGB codes, I've put those there to help you understand what's going on here. So for RGB, the minimum is 0 and the maximum is 255. So 128 is right in the middle. So this gray is right in the middle of each one. So it's important to understand that this gray has red, green, and blue in it. Okay. So when you're staring at red, these receptors, the cones in this part of your eye that code for red, they eventually get tired and they stop responding. They stop responding as much. They literally get tired. So you're removing the red part. And so what happens when you have red, your red's not firing, but all of a sudden you have your green and your blue firing? Well, that gives you something like Psyc. It's not, you didn't see, maybe it wasn't quite as brilliant as this because these two colors wouldn't be quite as high as 255. But the idea is that you'll see something like this because that's what you get when you have red, green, and blue and you remove the red. Down here it's the same story. You made green tired, and what do you get if you have red, no green, and blue? You get something like this. The same thing is true for yellow. Now this one is a little bit tougher to explain because it's not as straightforward. And the reason is that you don't have a yellow receptor. So yellow is already, in order to perceive yellow, you're already using multiple of your receptors anyway. So the story is a little bit, it's not as clean because it involves a little bit of red and green to begin with. Yeah?
Does this experiment work because like the screen's emitting light, or if you looked at a paper with the four colors, would you get the same effect?
Same. I mean, the only way you can see a paper with anything with color is that light is bouncing off of it. So either the light is bouncing off of some other source of light, bounce off the thing and then goes to your eye, or the thing itself is generating the light. Either way, it's the same. You have to have light going into your eye, but it would work the same no matter what. All right, so that's interaction across time. This is just a really obvious version of it. There's lots of ways in which this is true for your senses. But what you perceive at one point in time is influenced by what you perceive at previous times. It's not always just a clean slate. Making receptors tired or not tired is just the most sort of superficial way to demonstrate that point. But there's lots of different ways in which that is true for your senses. Another thing to get a sense of, and I'll just show it to you, is that what you perceive in one part of space is influenced by what's present in other parts of space. So this contrast example demonstrates that because those two bars in the center here, they're both exactly the same color of gray. So this bar is the same color as this bar. It doesn't matter what you can't. There's nothing you can do. No knowledge state is going to help you see them differently. You will perceive this as being as being not as bright as this. You can't not see it. No, it's not an expertise effect or anything like that. It has a lot to do with something we haven't talked about yet, which is lateral inhibition. So I don't want to go into that just yet. But the point I'm making here is just that what you perceive at this point in space is influenced by what your eye's doing, what light or non-light is going on at this part of space. Here's another weird example. If you look at these grids, you'll see something in the corners here. It's not there, but you'll see it, and you'll see it more if you're having a hard time seeing it. Just move your eyes from one to the next. As you move around, you should see something where there is nothing. This is also true. So you'll see areas of light in these corners, where even though there is none. You'll see areas of dark in these corners, even though there is. Okay, let's move on. So, what happens when the light comes into your eye, goes through all these cells, literally goes right through them. It influences some combination of rods and cones in your back of your eye, and then the signals go this way, and out of the ganglion cells into your brain. Well, where they go after that, the first thing to talk about is the fact that they cross over. So half the where's this, the bottom? So yeah, this is a little bit bizarre because it's upside down, but when light comes into your eye from the right side of left side of space, it's going to hit this side of your eye, right? That information is going to go to the right side of your brain. When information is in your right visual field, so over here, it's going to come in your eye, hit this side. Those receptors send their projections to the left side of your brain. So each of your eyes has a left and right visual field. So the left visual field of both of your eyes goes to your right brain, and the right visual field of both of your eyes goes to your left brain. After there, it goes to your thalamus, and in particular, I will talk about this in a second, the lateral geniculate nucleus. Just hold on for a second. But it goes to your thalamus, and then it goes to your primary visual cortex in the back of your brain, where most of the high-level visual processing, actually even low-level, the specific visual processing occurs. So one thing I wanted to point out here has to do with not only this. Yeah, I got it right here. Rods, cones, bipolar cells, ganglion cells. So actually, maybe you should go back to this picture so it makes sense. So you see how we've got these photoreceptors, but we've also got horizontal cells, these bipolar cells, and ganglion cells. One thing to keep in mind is that even though you've got 120 million rods and about 6 or 7 million cones, you do not end up with 127 million axons going out of the eye. So there's a lot of summary going on before the signal even leaves your eye. And in fact, it's by the time it gets to your visual cortex, it's reduced something around 100 to 1. And so you might have multiple bipolar cells. You might have one bipolar cell for multiple photoreceptors, and you might have one ganglion cell for a whole bunch of bipolar cells. So there's always going to be some sort of reduction that happens right here in your eye before it even leaves your eye. Oh, actually, I have the numbers here, right. So 125 million or so rosin cones. By the time you get to the ganglion cells, it's only about 10 million. And by the time you get, I'm sorry, the bipolar cells. And by the time you get to the ganglion cells, which are actually the ones that actually leave the eye, there's only about 1 million. So that's an insane amount of sort of summary. Why is it that way? Why not just send the whole 125 million to the brain? I don't know why it evolved that way, but it's probably a resource issue. It's probably just too much to process in your visual cortex, all that information. I don't really know. I'm just making that up. Okay, lateral vision. So we are running out of time. I'm just wondering if we should skip this part. I think I will. They talk about it in detail in your book, so I want to lock up. Okay, this is okay. I lock the whole thing up. All right, I want to fix this. All right. Well, while I'm rebooting my computer, let's talk a little bit about lateral inhibition. So you can talk about lateral inhibition on the single receptor level. Okay. So some receptors, when they get receptors are all. You saw that one microscope picture I showed you. All these rosin cones are really dense, really close together to each other. Okay. It's just like a carpet almost of receptors. When one receptor receives light and starts firing above baseline, in addition to sending that signal on, receptors can also, as a function of how much activation they're getting, they can actually use that activation to suppress the photoreceptors around them. Give me a second, typing this even longer constellation. Okay. All right. That's the right one. Yeah. So a single receptor, when it's firing, it can attempt to suppress the two. There's actually a whole range, but just to be simple, the two receptors beside it. Okay. Now, why would you have a system like that? Well, imagine you had a really tiny pinpoint of light, and you shine it at the back of your eye. Even the smallest one you can imagine seeing. It's the smallest light you can imagine from a flashlight. The smallest pin light is going to cover hundreds of receptors. Okay, but let's imagine it was just one receptor wide. So if you hit one receptor, it wouldn't matter. If that's how the world works, where every time light came into your eye, it was just one receptor wide, you would have no need for anything like lateral inhibition. But imagine it's two receptors wide. And so now it's going to come in and it's going to hit one receptor and it's going to maybe hit the receptors right around it as well. You want to have a system where a light that's hitting right on top of one receptor causes a lot of signal. But if it's just a little bit hitting on the receptors next to it, so a receptor that's only getting a tiny bit of light, you have a choice. You can either send that signal along and say, well, this signal is sort of on, this receptor is sort of on, and this receptor is really firing, but this one's just sort of on. That could be how it would evolve. We could have a system where all the data that comes in just gets sent right to your brain. But what happens instead is, if you have one receptor that gets light shown right at it, and the receptors next to it just get a little bit, instead of sending, again, let's just pretend there's three. So me, the central one and the two beside it. Instead of sending three signals, one that's all the way to 11 and the other two that are at a 2, your eye just sends one signal. It just, because of the lateral inhibition, the one that gets the most signal, it has the most inhibitory power to inhibit the ones around it. And that's the way that we get sharp, even though the world is fuzzy and light doesn't always hit exactly in one spot, and sometimes light bleeds over even though the source really isn't in that location of space, we end up with clear vision because it's lateral inhibition. Now, most of the time, the lateral inhibition is not one receptor and two other receptors around it. What you actually end up getting is these big collections. I'm sorry, that can happen, but mostly you get these big collections of receptors. And we usually think of them as sort of a circle, and there's a big circle and there's a little circle. And they're all just photoreceptors, but there's a big circle and a little circle, and they can either be arranged in a center on off surround or a center off on surround. And so again, what that means is if light comes into the center of this circle and hits the internal circle, then the receptors there will suppress the ones around it. There are other of these. There's other of these sort of collections of receptors where sometimes when the light hits the center, nothing happens. But if it's the edges, then those receptors will fire. And those big collections of receptors are usually eventually connected to a relatively small number of ganglion cells. Let's just, for simplicity's sake, let's say an entire field like this is connected to one ganglion cell. And so what that means is that group of receptors is only going to fire if the light hits either right in the middle or doesn't hit in the middle, depending on whether it says on surround or off surround. And so you have these different organizations of receptors in a scheme to simplify the data that's coming in. So ultimately, you want you only want the most clear signals to cause changes in the back of your eye, and that's what this lateral inhibition is. Your book talks about this and has lots of pretty graphs, and so please read that as you normally would. But just keep in mind that the reason I want you to know about it is not because the details of lateral inhibition are important here. What's important is that I want you to have some idea of the way in which your eye takes 127 million potential points of light and reduces it down to just 1 million. Great deals of summary have to go on, and the gist is that your brain really just wants to receive the clearest possible signals. What is the clearest summary of the current view that can be given? That's what this summary is about. All right, that's all we can do for today.